Welcome to the video for chapter 7.1. We are going to be talking about energy and how we make energy in living things throughout all of chapter 7. So chapter 7.1, we're going to talk about some basics of energy production and some terminology that we need to know and how this is going to apply to us in microbiology. Okay, so our chapter 7 objectives, because chapter 7 is all about bacterial metabolism, we need to be able to describe the various kinds of biochemical reactions that provide energy for life. We need to be able to explain ways that bacterial metabolism interacts with human health. We need to be able to describe how microbial photosynthesis and nitrogen fixation support life and how these processes can affect human health. Okay, so we're going to talk about a lot about diagnostics and some of the metabolic pathways that we harness to do that. So let's talk about some vocabulary we should know. Okay, so this might look familiar if you took Biology 201. This is Biology 201's metabolism slide. So just a reminder, metabolism is literally all of the chemical reactions that is happening in a cell or organism. Okay, whereas a metabolic pathway is going to be part of that metabolism. It's going to be a series of biochemical reactions that are going to take one or more substrates, so one or more reactants, and it's going to go through a series of reactions where it takes that initial substrate, that initial reactant, and makes it into an overall final product. Okay, so what we're literally going to be doing here in chapter 7 is talking about a couple different metabolic pathways. Okay, so when we are talking about different metabolic pathways, we're really talking about two types of metabolism, and we need to know these definitions. So we have catabolism and anabolism. So catabolism is when we have energy releasing reactions, and anabolism is when we have energy capturing or energy requiring reactions. So the thing about catabolism and anabolism, they're always linked together. They're what we call coupled reactions. So we can't have one without the other. So we usually discuss them in terms of individual reactions and what is happening. I want you to know these definitions. So an anabolic reaction is going to require energy, and it's going to take small molecules and it's going to build them up into bigger molecules. Okay, so the reason why anabolic reactions require energy is because we're forming bonds. Whenever we're making bonds in between things, we're going to have to put energy in to make that bond. So anabolic is going to require energy and build things. Catabolic reactions are going to break bonds, so they're going to release energy. So they're going to break down large molecules into smaller molecules, which is what's going to provide the energy. You need to know the difference between these two, and you need to be able to tell them apart. Now, this is going to sound a little bit silly, but one of the ways I remember catabolism versus anabolic, we're going to go with anabolic. That was hard for me to say. So the way I remember catabolic versus anabolic is cats. If you have a cat, maybe we had a cat when I was growing up. Cute little guy, pain in the butt little guy. Our cat's favorite thing to do is any glass of substance, water, soda, anything. If you left it unattended on the counter, on the table, on the floor, it was our cat's life mission to knock that cup off and break the cup and spill everything. So I remember catabolic is like the cats, like our cat. They like to break things. So catabolic breaks things. Okay, and they release energy and run away because they're going to be in trouble. It's a little bit silly, but this is how I've remembered it 20 years on. Okay, so we need to know the difference between these two types. Okay, so we're at the beginning of a new chapter, which means we have a new case study to talk about. These are always fun. So chapter 7's case study is called A Spring Break Mishap. Oh, this one's gonna be good, I can tell already. Okay, so let's set the scene. So we have Shane. He's a 21-year-old college student. He's away on spring break, no class, no work. He's just having fun. So like a bunch of other college students on spring break, Shane is in Cancun, Mexico, enjoying the beaches, enjoying some sunshine, also enjoying some alcoholic beverages from the beachfront stands. Hey, guys, what's the number one rule when you're in Mexico? We never drink the water. Let's keep that in mind. Okay, so Shane has a wonderful spring break. He drinks a lot, he plays a lot, gets a lot of sunshine, gets a nice tan developed, has so much fun, and then he goes back to school at the end of spring break. But the problem is shortly after Shane gets home, he starts to get really sick. He starts to have some really bad stomach cramps. They're hurting him. He has watery diarrhea. He has a fever set in after he starts getting these abdominal cramps and this diarrhea. His fever's really high. It's about 102 degrees Fahrenheit or 38.9 degrees Celsius. So he's just not feeling great. So thinking, well, he's been traveling, he's gone through airports, he's been through a lot of people. He's starting to think that maybe he just got a flu bug. So he just kind of hunkers into his apartment and waits for it to pass. Well, if it had been a flu virus, his method of getting better would have probably worked. But the thing is the very next day, Shane still has diarrhea, but he's noticed that he has blood in his diarrhea. He has blood in his stool. This is never a good sign. In fact, when you see blood in your stool, this is what we call dysentery. So if you ever played the old Oregon Trail game, most of us died of dysentery, usually because we drank bad water on the Oregon Trail. I usually died of that or snake bite. Okay, I never got one of the really cool ones like getting gored by a bison or something. Okay, so Shane has dysentery. We all know blood in our stool samples are in our stool. It's really bad juju. It's really bad. It's just bad. So Shane, knowing it was bad, got himself up, got himself presentable, and then took himself to the college health clinic. He got to the health clinic, told them what was going on. They saw the amount of blood in his stool, and they're like, we cannot help you. We are not equipped for this. So they automatically sent Shane to the local ER where he was admitted to the local hospital. Okay, so when he's in, he tells the attending physician all of his symptoms, what's going on. They notice that Shane is very obviously dehydrated from the diarrhea. We talk a lot about diarrhea and dehydration in this class. His stool sample is very, very bloody. And one of the things that they did is, once they saw the blood in his stool, they gave Shane a rectal exam, and there were some problems with this rectal exam. It was very painful to Shane, and it caused bleeding, which is never a good thing. Okay, well, obviously something's going on, so they're going to take a sample of Shane's stool. Now, if you're a medical microbiologist, you deal with a lot of crap, a lot of poop. This would not be the funnest job ever. Okay, so they take Shane's stool sample and they're going to start to run a bunch of tests on it. Well, because he has diarrhea and stomach cramps, they're starting to think that it's probably an enteric or intestinal bug that's causing all the problems, which just means it's probably going to be a gram negative. So with the tests that they're going to choose to run, they're going to want to run some selective, meaning we want to prohibit growth of a certain type of bacteria, and differential bacteria in a differential test. So they're going to use what is called Hectoan agar. We've talked about this in class. We've seen it a little bit in lab. Okay, so what Hectoan agar does is it's selective. It only allows gram negatives to grow, and it has bile salts in it that's going to prevent the growth of gram positive cells. Okay, so it's also differential besides selective, and it's going to help us differentiate between different types of bacteria. Now, we can tell the difference between a bunch of different enteric gram negatives. So there's Salmonella, there's E. coli, there's Shigella, and Cabecilla. I can never see that one. We'll see it on a slide in the few slides. Okay, so we need to be able to tell the difference. Okay, so these bacteria go through different pathways. So one of the things that we see is Salmonella is when it starts to go through its metabolic pathway, it's going to produce hydrogen sulfide, okay, which is going to take the colony on the hectoan agar and it's going to turn it black because of this sulfur that's contained in this hydrogen sulfide. Okay, so that's one way we can tell that from the rest of them. So both Shigella and E. coli are going to produce colonies that are not black. So depending on what's happening, depends on what color you're going to get. So with Shigella, it doesn't ferment sugar and it doesn't make hydrogen sulfide. So it's going to stay its regular color. So it's going to appear to be a pale green colony, the same as the agar plate's color. Okay, with E. coli, and the other species that we can differentiate on it, they are sugar fermenters. They're not going to make the hydrogen sulfide like Salmonella. So what they're going to do is they're going to ferment sugar and they're going to become this pink color. Okay, so the lab grows Shane's culture and he produces colonies without color. So that eliminates E. coli and the other bacterial strain and Salmonella. Okay, so what Shane's hectoan plate shows is that he probably has the Shigella pathogen. Okay, so with the results of Shane's hectoan agar plate, what they did is they took the bacteria and they did what is called serotyping with it. So they used different antibodies and reacted to them with the pathogen to determine what specific strain of Shigella Shane had. So the serotyping confirmed that Shane has Shigella flexneri. So Shigella flexneri causes common traveler's diarrhea, so it causes dysentery from drinking contaminated water. So when Shane was on spring break, he was having all those drinks at the beachfront stands. So maybe they're trying to make more money and they watered the alcohol down, or he got it on the rocks, meaning he had ice, which was made with the local contaminated water. It doesn't really matter what happened. This is probably where Shane got his infection and got the disease. Now, the thing with Shigella is really interesting. It has what is called a low infectious dose. If you remember back from a couple chapters, this means we don't need a lot of bacterial cells in order to cause disease. So with Shigella, you only need like 10 bacterial cells in order to start to see symptoms and to get this disease and to become sick. So a lot of times when you're in places like Mexico or in South America where they have a lot of contaminated water, one of the things you do is you always use bottled water to do everything, even simple things like brushing your teeth. Because Shigella flexneri doesn't need a lot of bacteria in order to cause disease, just brushing your teeth with contaminated water, so using the tap water, is enough to give you this infection. So whenever you're in Mexico, make sure you're using the bottled water instead of using the tap. Okay, so even swishing to get the toothpaste out of your mouth is kind of dangerous this way because that can introduce at least 10 bacterial cells to your mouth. Okay, so because he's severely dehydrated, Shane's going to get IV rehydration therapy. And one of the things they're going to do is they're going to run a series of tests based on the specific strain of Shigella that Shane has. So they're going to grow it on a plate, and they're going to use little antibiotic discs in order to see which antibiotic works the best on this pathogen. So they're going to test the pathogen's antibiotic sensitivity. So they put a bunch of different antibiotic discs and they go to see which one kills the most bacteria around it. Fun note, we are going to actually do this in the lab. We're going to use these antibiotic discs to figure this out. So Shane's antibiotic tests show that the antibiotic quinolone was the best one to get rid of this infection, so that's what they gave him. Okay, so it took a few weeks, but Shane made a full recovery. Okay, so let's look about what a hectoan agar plate can look like. Okay, so we can differentiate between 4 types of bacteria, so we can identify 3 different groups. Okay, so we have lactose fermenters like E. coli and Klebsiella. Sorry guys, that one's hard. Okay, since they both are sugar fermenters, they're going to turn pink because of the production of lactic acid. You have the salmonella that's going to create black, like this little guy here, black colonies because it's producing that hydrogen sulfide. And then the Shigella is not going to ferment anything. It's not going to make hydrogen sulfide. So it's going to stay the green color of the agar. So this is what you see with Shane's diagnosis. Okay, so that's one way we use metabolism as a diagnostic tool. In fact, most of our selective and differential plates are going to lean into these different metabolic properties that bacteria have in order to tell them apart. Okay, which leads us to chapter 7.1, where we talk about energy for life. So section objectives, we need to be able to define the major classes of energy yielding metabolism, including photosynthesis, organotrophy, and lithotrophy. Okay, you need to be able to explain the importance of energy and entropy for living cells. You need to be able to state the role of enzymes in controlling metabolic biochemistry. So we have a pretty fun analogy to help us with that last one from Dr. Burrows. Okay, so let's talk about cells. Cells are highly organized. So one of the ways that organisms get highly organized cells is they're going to take smaller molecules like these guys, so ammonia and iron 2 plus and carbon dioxide, ethanol, magnesium 2 plus. They're going to take this and they're going to take these smaller molecules and they're going to combine them to make more complex structures through a process called biosynthesis. So whenever we take things that are small molecules and we use them to build bigger things, we have to use energy in order to bring these things together, right? We have to be able to form the bonds. Okay, so this process is called biosynthesis. Biosynthesis makes biomass. So biomass is just the organic matter or the organic compounds that are made during these processes. So biomass is what a lot of things like us use to get energy. Okay, so when we're talking about energy and we're talking about organization, we have to talk about something called entropy. You know, one of my favorite words. Remember, entropy is the measure of chaos in a system. Okay, so if we're becoming more organized, okay, so if we're going from small things to large things, we're becoming more organized. If we're becoming more organized, there's less chaos. So entropy is decreasing as we become more organized. Okay, so we have to talk about how energy flows through the biosphere. In chapter 6, we talked about how all energy comes from the sun and it goes to autotrophs and then to heterotrophs. This is kind of what this slide is talking about. We need to think about the terms of taking that energy from the sun and making it usable is going to be some sort of organism's metabolism. Okay, so we have the sun's energy coming in to Earth. Okay, we have our photoautotrophs, our autotrophs that do photosynthesis. Okay, they're going to take this energy from light and start using it to make biomass, make these organic compounds like glucose. Okay, so when energy is gained by Earth's biosphere, that means we're increasing the order, which means we're decreasing the entropy. Okay, so as we start to make these different sugars, okay, as we start to make biomass, we are going to always lose some energy in the process to heat. Okay, it's going to become unusable. So this heat loss is what we also call entropy. Okay, because as we lose heat, we're going to add chaos to the system around the molecules being made, which is going to increase the entropy. Okay, so we have the heterotrophs, which are going to take the sugars made by the autotrophs, and they're going to start to break them down to make energy. We're going to lose a little bit of energy to heat. Order is going to decrease. Entropy is going to increase. Okay, so we're going to use this biomass, these organic compounds, to fuel us. We also make biomass. Then there's a third category of individuals called decomposers, which are going to take the dead organic matter and use it to make energy, and it's going to just flow through the system. Okay, so this is a good graphic to know and understand. The thing is, out of these three, photosynthesis is going to be the most efficient process to produce energy. But the thing is, even our most efficient process can't be 100% efficient. Okay, we're always going to lose a little bit of energy due to heat loss, due to that entropy. So with photosynthesis, we lose a little bit less than we do with heterotrophy and decomposition, but we still lose some nonetheless. Okay, real quick, let me just remind you about entropy. It's the measure of randomness or disorder in a system. Okay, so the more ordered something is, the less entropy it has. Okay, the more chaotic a system is, the more entropy it has. So if we think about the different states of matter, they have different levels of entropy, where solid has the least amount of entropy and gases have the most. So as we go from solid to liquid, we increase in entropy, and as we go from liquid to gas, we increase in entropy. Okay, and this is all because we are losing heat when we increase the energy in the systems that are going to cause it to move more, to be more chaotic. So entropy is going to increase as disorder or chaos increases. Okay, so we have another little table. These are going to be different metabolic reactions that yield energy. We've talked a lot in biology about photosynthesis, so phototrophy. So when we're going to take light energy and we're going to use it to make high energy molecules that donate electrons to acceptors. Now, one of the things I want you to start thinking about when we talk about metabolism, and we start talking about things like high energy electrons are just another way to say this is what's going to provide energy to do other reactions. So high energy electrons are the energy to do other reactions. Okay, so we have phototrophy, which we're going to harness the energy from the sun, and we're going to put that into the energy from the sun into high energy electrons that are going to then go to take 6 waters and 6 CO2s, and it's going to combine them to make 1 molecule of sugar and a waste product that's very near and dear to our hearts, oxygen. Okay, so phototrophy, that means they're autotrophs. They're going to make their own food from light. The rest of us are chemotrophs, right? We are going to use food to get our high energy electrons. Now, from where we get our food, that's going to depend on what high energy electrons we get and how efficient the energy making process we go through. Okay, so we are aerobic organotrophs, which means we are going to use an organic molecule like glucose, and we're going to take its high energy electrons and we're going to donate them to oxygen. So oxygen is our final electron receptor. That's what it means here. Okay, so we're going to break sugar down with oxygen to make water and CO2 and a whole bunch of energy. Okay, so that's aerobic organotrophy. So we have anaerobic organotrophy. So this is similar to aerobic, except the organic molecule that we're going to break down is going to donate electrons to either itself again or another molecule. Okay, not oxygen. Okay, so fermentation, which happens in anaerobic environments with no oxygen, fermentation is an example of anaerobic organotrophy. Okay, we have anaerobic respiration, where we can donate to an inorganic molecule like sulfur. Okay, we'll talk about that later on in this chapter. So with fermentation, we're going to take that sugar that we got from our food molecules, and we're going to break it down into ethanol and carbon dioxide. Okay, so we're not going to donate our high energy electrons to oxygen in anaerobic organotrophy. Okay, so that brings us to lithotrophy. We have aerobic and anaerobic lithotrophy as well. Okay, in aerobic lithotrophy, we're going to have an inorganic molecule that's going to donate electrons to oxygen. So this is going to be things like ammonia, NH3, donating electrons to oxygen to make nitrate and water. We have anaerobic lithotrophy, which is going to take an inorganic molecule and it's going to donate its electrons to another inorganic molecule a lot, something that's not oxygen. So a lot of times in anaerobic lithotrophy, we're going to have an inorganic molecule like carbon dioxide donate its electrons to something like hydrogen gas, and we're going to go through something called methogenesis, which is going to be the production of methane and water. Now, with lithotrophy, there's a really interesting thing that happens, especially to farmers. Lithotrophy comes to the forefront a lot when farmers overuse nitrogen fertilizer. So like they high spread manure or they use too much synthetic fertilizer. So what happens here is those nitrogen containing fertilizers contain ammonia. So the lithotrophs in the soil are going to take this ammonia that's now in abundance and it's going to start to convert it to nitrates. Okay, so it's going to lead to high levels of nitrate production, which are a byproduct of this lithotrophy pathway. So we're going to have all these nitrates that are going to build up and build up in the soil, and it's going to actually cause the nitrates to run off into the local water or the nearby water sources, which can then cause downstream from where the runoff is happening, algal blooms. So we're going to get a lot of algae in the water, which in itself cause a lot of problems. Okay, so there's definitely some pros and cons to each of these different methods of producing high energy electrons, but they all are going to be useful to making energy. And we're going to talk about these as we go through chapter 7. Okay, so when we talk about metabolism is all the chemical reactions in the body. But these chemical reactions are just not happening willy-nilly. They are actually pretty controlled. In fact, proteins called enzymes are going to control these reactions. So with energy, we have the law of conservation of energy, which means that energy cannot be created nor destroyed. It can be transferred and transformed. Okay, so the energy in living systems is constantly being recycled and repurposed. Okay, so enzymes are going to monitor this energy recycling and repurposing. Okay, so it's going to monitor the reactions that are happening. So if you remember, enzymes are going to lower the activation energy of chemical reactions so they can proceed, but they're also going to make sure they don't go too slow, because if they go too slow, life dies, and they're going to prevent them from going too fast, which has some detrimental effects as well. Okay, so these enzymes are going to control catabolism and anabolism. Okay, so for an example, we go through sugar catabolism. We're going to break sugar down to release energy, which then is coupled to an anabolic reaction where it's going to require energy. So our cells are going to take the energy from breaking sugar down and they're going to use it to make proteins, to synthesize proteins. Okay, so enzymes are going to manage this energy transfer from one reaction to another so we have enough energy to make things and to break things down. Okay, so let's talk about an analogy. Now, I'm a little bit upset I didn't think about this. This is an excellent analogy. All the credit goes to Dr. Burrows for this analogy. So we have to talk about marshmallows. Okay, we are like marshmallows. Some of us are soft and fluffy, but we're all full of high energy nutrients. Okay, so I want you to think about this. We've all put marshmallows on a stick. We've all put marshmallows in a campfire. So what happens when you start roasting your marshmallow? Now, it depends on what type of marshmallow person you are and how much patience you have, right? So my best friend, super patient, loves a nice golden brown marshmallow, so he will slowly rotate that marshmallow over the fire. He's very controlled about it. I So I don't like marshmallows. I do like roasting marshmallows on the fire. Like I will roast marshmallows for people, specifically my grandmother. So my grandmother is kind of an anomaly with marshmallow roasters. She doesn't like a brown marshmallow. She likes hers black, which is always great because I do not have enough patience to roast a golden brown marshmallow for her. So for my method of marshmallow roasting, my marshmallow goes really close to the flame. It catches on fire pretty quickly and starts burning. This is actually a uncontrolled chemical reaction. Combustion's uncontrolled. So this is one of the things that in your bodies enzymes are trying to regulate. Okay, because you are a lot like this marshmallow. You have a lot of these nutrients. You have a lot of sugar. So the reactions in your body, if they proceed too quickly, you're actually going to build up a lot of energy. That energy is going to have to go somewhere. It's going to ignite, it's going to combust. So you will literally light on fire or burst into flames because of all that energy stored in you that you're now proceeding to need to use in this chemical reaction. Okay, so enzymes are going to make it so you literally don't combust. So they're going to harness these reactions, and they're going to make little bits of energy at a time that can be used to make other things, and you don't make too much at a time and combust yourself. You go through spontaneous combustion. Okay, so enzymes are really, really important to control these reactions. Okay, so sugar metabolism, this is going to be the main thing we talk about in chapter 7. Not everything goes through sugar metabolism, but a lot of bacteria and we as well go through sugar metabolism. This is sometimes called glucose catabolism or sugar catabolism. Okay, so we're going to take an inorganic carbon and water. If we are autotrophs, we're going to harness solar energy to go through photosynthesis to make glucose and oxygen. Now, for us, we do the opposite reaction. So we take oxygen and glucose, and we take the energy from glucose and give it to oxygen. And through a series of pathways, we're going to break it down to water and carbon dioxide and make ATP and some heat that we lose. Okay, this is going to be the whole focus, or most of the focus of chapter 7 is talking about both of these reactions, and we'll throw in some lithotrophy there. Now with cellular respiration, we can make this sugar yield 36 to 38 molecules of ATP, which is quite a lot of energy, and that's going to be for aerobic respiration. But this converse reaction, it's going to take 18 molecules of ATP, okay, to make 1 molecule of glucose. We also use a molecule called NADPH. You can think of 1 NADPH is equal to 3 ATP molecules energy-wise. So we're going to not only take 18 ATP molecules, we're going to use about 12 NADPH molecules, which is going to put all the energy into that molecule of glucose that's being made in photosynthesis. This is why sugars are really great sources of energy. This is we can break them down into all that energy that we put into them. So that is chapter 7.1. Just a reminder, all energy on Earth comes from the sun. Different types of metabolism are going to yield different amounts of energy for different microbes. The important ones to know are phototrophy, organotrophy, both aerobic and anaerobic, and lithotrophy, both aerobic and anaerobic. And we need to remember that enzymes are the things that are controlling all these reactions so they don't get too out of control or they don't go too slow. Okay, so that's all for chapter 7.1. We will see you in the lecture for 7.2.